And now we are going to see the processes involved in the particular genetic engineering. So as I told you, our aim over here, we have to target the required gene and we have to isolate the required gene. Then we have to isolate the particular vectors. We have to cut both the fragments. Then we have to introduce the isolated gene into the particular vector. Then we have to transfer this created rDNA molecule into the host. And in the host, we have to multiply the particular host cell along with the products. Then we have to harvest the product, purification of product, then commercialize the product. These are all the steps we have seen till now, and now we are going to see the details of each and every step. So what is the first step? As I told you, the isolation of the total genomic DNA. So for example, you need a protein called insulin. Okay, that is what we are going to discuss in the next chapter. You need a product from insulin of human gene. Okay. So what you have to do, first you have to isolate the gene which codes for a product insulin. So where it is in a human body. Okay. So in a human body, all cells, they are not going to express the particular insulin. There are some cells in the particular organ, and there are some chromosomes in the particular cells, and there are some locus or alleles in the particular chromosome. They are going to express the particular gene or product. Okay, so for a specific product, you have to locate and isolate the particular specific region of the particular entire genome. But practically it is not possible to locate a specific region in the mixture. For example, in case of human being, there are around 40,000 types of genes in a single cell. So if you are going for a single gene isolation from a mixture, this is not practically possible. You cannot search for a single gene in the mixture of 40,000 different types of genes. Each and every genes are in number of copies, thousands of lakhs of copies. From the trillions of copies of genes, you cannot pick a single copy of your required gene. So that you have to go for the entire or total genomic isolation. Okay, that is what the first step in the the isolation of the total genome DNA. So based on a source, the steps may vary. Okay, for example, if you are going for bacteria, you have to lyse the cells by means of rupturing the bacterial cell wall that is made up of the peptidoglycan. So normally in case of bacteria, the bacterial cell wall is made up of peptidoglycan. So you have to go for enzyme lysozyme. Lysozyme is an enzyme which can digest the glycosidic bonds in the cell walls of the bacteria. And in case of fungi, in case of plant cells, in case of fungi, the cell wall is made up of the chitin. In case of plant cells, they are made up of cellulose. In case of animal cells, there are no cell wall. Okay, so based on a cell type, you have to choose the rupturing method of the cells. So whatever may be the cell, as I told you, you have to break open the cells, then only you can explore the internal molecule, that is the DNA of your interest. Okay, so whatever may be the cell type, based on a cell type, you have to choose for enzymatic treatment, and by the enzymatic treatment, you are going to open up the cells and that will expose the cell membrane. Then by means of some chemicals, for example, NaOH, sodium hydroxide, SBS, EDTA, tris. Okay, by means of some mixture of enzymes, we are going to rupture the cell membrane. Then we will get into the internal regions of the particular cells. That is what called the cellular extract. And within the extract, there are so many molecules available: proteins, nucleic acids, other RNA molecules, and the plasmid DNA molecules, all the lipid molecules, carbohydrates. So what is the target molecule for you? Only the DNA molecule. So you have to eliminate all the remaining molecules. That is, we have to eliminate the proteins, we have to eliminate the RNAs and other nucleic acid molecules. So we have to use specific kind of enzymes called RNases to digest the RNA molecules, proteases to digest the protein molecules. Then in the mixture, you are having only the DNA as an intact molecule. Then you are going for repeated centrifugation, and at the end, by means of the ethanol, the ice cold ethanol, we can precipitate out the DNA molecule. This is what the steps involved in the isolation of the genomic DNA from any source, whatever may be the source. The initial steps may vary based on a cell type. The cell wall digestion by means of enzyme may get vary, but whatever may be the cell method, the ultimate aim is we have to open up the cell, we have to break the cell membrane, we have to get into the cell extract, then we have to digest the unwanted proteins and RNA molecules, then we have to purify the DNA, then we have to finally precipitate off the DNA by means of the ice cold ethanol. This is what the steps involved in the isolation of genomic DNA. Then, so the next step, as we have discussed, the cutting and joining, the manipulation of the DNA molecules. Okay, so manipulation we have discussed the detailed in a previous section. I will repeat it once again. There are some enzymes called restriction endonucleases, and there are some enzymes called ligases. With the restriction endonucleases, with their specificity, we can cut the DNA at a specific location in the mixture, and that will make a required fragment of a required gene. And with the same enzyme, we are going to cut the plasmid or carrier molecule, so that we are getting the complementary bases at the sticky ends, and then we are going to join the particular required gene, or we are going to insert the required gene into the particular plasmid, and that will create a recombinant DNA molecule. This is what the steps involved in the cutting and the joining. I will repeat it once again. This is the genomic DNA, just imagine, and from this genomic DNA you have isolated a fragment. Okay, then you have digested the particular fragment, so that you are getting you are going to get the fragment like this. Okay, it should be complementary to each other, it should be sticky like this. Okay, so you are gonna get this is your gene of interest, and at the same time you have isolated your plasmid molecule, and you have digested the plasmid with the same enzyme, so that the plasmid also they are going to produce the protruding end like this. Okay, so now you are going to mix your gene with your plasmid. This gene will be inserted into the particular plasmid or carrier molecule. Now you have a recombinant DNA molecule, that is, it carries your required gene, and this molecule is called rDNA or recombinant DNA molecule. Then next step is we are going to introduce the particular recombinant DNA molecule into the host cell. These are all the steps involved in the cutting and joining. Cutting by means of restriction endonuclease, joining by means of enzyme ligases. Fine. And before we are entering into the section called the delivery of rDNA into the host, you should know the technique called PCR. So as I told you, if you have a very low copy number or low copy of genes, you can amplify your required gene by means of process. Okay, amplification or increasing in the number of copies by means of process called PCR. PCR refers to polymerase chain reaction. Okay, polymerase chain reaction. This is a technique. With this technique we can manipulate, or we can multiply the number of copies of a required region of a particular gene. This is what the thing you have to remember. Over here, if this is your required gene, okay, so there are so many basic requirements we need to proceed with the polymerase chain reaction or PCR. The first thing, you require primers. Okay, specific primers. For example, if this is the entire length of DNA and you need a specific region, then you need something, some small oligonucleotides that is complementary to the ends of your required gene. Okay, so that is what called the primers. There are some small nucleotides. It will be around 10 to 12 nucleotide length. 10 to 12 nucleotide length. That is what called primers. They are complementary to the ends of your required or target gene. And then you need a major group of enzyme called DNA polymerase. Over here, the PCR reaction involved some high temperature. Okay, so you need some thermostable enzymes, the enzyme which can stable at high temperature. So we are isolating the enzyme from the thermophilic bacteria, that is the enzyme Taq polymerase. And for the polymerization reaction, you need the substrate or individual monomers, that is called deoxynucleotides. Okay, DNTPs, deoxynucleotides, all the deoxynucleotides, D, ATPs, TTPs, CTPs and GTPs. This is what called deoxyribonucleotides. Fine. And these are all the major requirement for any PCR. So what we are going to perform over here, PCR is a mission. Actually it is a mission. So what it will do, we are going to give a program to run the particular mission. So actually in a point of tube we are going to perform the particular reaction. In the reaction mixture, the mixture will contain the primers, enzymes, the DNTPs along with some buffer. So what will happen over here within the particular mixture? Okay, we are going to give the program, as I told you, the programs in a cycle. Okay, in the first cycle the DNA will be, okay, in the first cycle and the first step. The first step, the DNA will become a 2 single stranded. The double stranded DNA will become 2 single stranded by means of the increased temperature. Okay, by increasing the temperature it will break the hydrogen bonds in between the 2 strands so that it will become single strands. Then we are gonna immediately reduce the temperature. For example, in the first stage we are going to increase the temperature up to 90 degree. Then we are gonna reduce the temperature to something around 60 degree. Okay, so what will happen? Automatically we try to re-anneal. In that stage the primers will form annealing to the particular ends of the single stranded molecule. Okay, and then we are gonna give for the next stage, that is called the polymerization. In that stage the Taq polymerase will use the DNTPs and that will copy the required gene like this. The polymerization, synthesis of the complementary strand by using the template strand. And again, so now your required gene is being copied by the primers with the help of enzyme in the particular reaction mixture. This is the first cycle. Okay, this first cycle will take some few minutes, okay, few seconds to few minutes. Okay, so we are going to give some 3 hours to 4 hours for the cycles. Normally we will go for 20 to 25 cycles. So what will happen after 20 to 25 cycles? Only the required gene, okay, only the required gene will get amplified again and again and again and again. So after some 25 cycles you will have some millions of copies of your required gene in the particular point of tube, in the particular PCR mission. This is what the steps involved behind the particular polymerase chain reaction. I will repeat the steps. Polymerase chain reaction, the aim over here is the amplification of the required or specific location of your gene. So over here we are going to use the primers. Primers are nothing but small nucleotide sequence. They are complementary to the ends of the required gene, and we are going to use the enzyme Taq polymerase, and we are going to add the monomers of deoxyribonucleotides, and we are going to put it into the mission called PCR. In the mission we are going to set up a program with 20 to 25 cycle. Each cycle contains some steps, and in the first step the DNA will be separated out into single strand. The second step, the particular primer gonna join with the or anneal with the or gonna complementary base with the particular single strand, and the third step, the polymerase gonna synthesize the complementary strands, and in that way the cycles will continue, and after the completion of the entire program the required gene will be multiplied or amplified into huge trillions of numbers. In that way you will get huge volume of your required gene to go for the manipulation with the genetic engineering. Fine. So now we have, as I told you in the previous stage, we have prepared the required DNA along with the vector that is called the rDNA, that is the prepared DNA along which carries your required gene. And what is the next step? You have to introduce into the bacterium, okay, by means of process called transformation that we have seen in the previous steps. Fine. So by means of selectable markers, as I told you, antibiotic resistant markers or some other markers, we can select the correct bacteria. That is what the thing you have to remember. Correct. So what do you mean by correct bacteria? This is a bacteria which contains the plasmid, that plasmid which carries your required gene. Okay, that is what called the correct bacteria. Okay, how can we find out the correct bacteria? By means of selectable marker that is being carried out by the plasmids. Fine. So by choosing the correct bacteria, then we are going for the production. So over here in the production stage, what we are going to do, the actual steps involved in the microbiology lab. So we are going to inoculate the particular bacteria into huge bioreactors. Bioreactors, as we have discussed in the previous chapter, the fermenters. Okay, so in the huge fermenter we are going to give some raw substrate or the nutrients to the particular bacteria. Then we are going to inoculate the particular bacterium into the particular substrate, and in the particular substrate that bacteria will digest the particular nutrient medium, then that will produce the products. So the point you have to remember over here, the bacteria which carries the plasmid which carries the gene of your interest. Okay, the genes, it will expressed into product. Okay, in a suitable condition the genes will be expressed into product, and we are gonna produce huge amount of product within a bioreactors or fermenters. And now you have huge volumes, some gallons of products in the particular fermenter or bioreactor. The final stage, the downstream process. So what do you mean by downstream process? The purification of product. Purification of the particular product from the particular mixture. Okay, so what are all the things you have to consider in the downstream process? We are going to discuss the details of downstream process later in the future classes, but the point, the major steps involved the downstream process. This is the fermenter which carries the product. Where is the product? The product is within the bacteria. Where are the bacteria present? The bacteria are present along with the nutrient material and also the waste. Okay, so from the particular mixture, from the particular fermenter or bioreactor, you have to target and isolate your required product. So what are the steps you have to consider over here? First, you have to eliminate all the unwanted nutrient materials and the nutrient waste, the metabolic waste from the particular bioreactors. That is the very first step. That is, you have to separate out only the bacterial cells from the mixture. That is the very first step. Then now you have the bacteria which carries the products. Within the bacteria your product is being filled. Now again from the bacteria you have to purify your product. Now you won't need the particular bacteria, so you have to kill all the bacteria, you have to break open the bacteria. Now the internal areas, that is the extracts of the cells available, the extract which contains your protein of your interest, that is product of your interest. Now you have to go for protein purification. There are so many techniques available. There are some chromatography techniques available, centrifugation. Okay, and there are so many techniques available to purify the particular protein product, and finally the commercial product is ready to commercialize into the market. Okay, these are all the processes or steps involved in the biotechnology or making of a final product. And now with this we are going to complete the particular chapter, and the next chapter we are going to see how can we use this technique to manipulate our industrial application or the commercial or human society application. What may be the applications of this technique to agricultural industry and as well as in the human or health industry. That is what we are going to discuss now.